All right, hi everybody. Let me know if you can't hear me. Thanks for coming, everybody. That's virtual. We have a couple people online and a couple folks in the building here. We're just going to get going. Hi everybody. My name is Sam Beck-Anderson. I'm the Associate Director of the Finger Lakes Institute and the coordinator for the Finger Lakes Partnership for Regional Invasive Species Management. I appreciate everybody who's taking the time to come out today. I think we should have a little bit more folks coming later in the afternoon for our macrophyte survey program training. But this is a good group of partners here to just generally talk about what we have going on. There is a lot, ultimately we were a little bit late on kind of some of the organization of this. So like I said, I appreciate everybody taking the time to come out and everybody's interest and making this a priority. I'm hoping that you get something out of this today. So just an overview of what we're going to talk about. I'm just going to be getting, for reasons that will become clear soon, I will be giving our program updates for the year, sort of what we've been able to achieve in the past several months since the last time we met with our partners and what we have planned for the field season. We're also going to be welcoming our own Josh Neff to talk about a more in-depth description and report on one of our kind of interesting fun projects that we've been working on for a couple of years now, which I'm excited to hear. We are going to open the floor to partner updates, and if we can't fill that time because we have a limited group, then we'll just sort of take a little break before we hit the macrophyte survey program training later on. So with that, I'm just going to kind of jump right into my first presentation here. Okay. So as we have had for several years, we're going to be leading an aquatic strike team this summer again. This year, I guess last year, we made some significant changes in our scope as well as added a lot of or modified the way that we're collecting data for our AIS strike team. We are limiting it to specific water bodies and reducing the overall sites in preference of revisiting locations over the course of a season. So we will be doing monthly site visits to these locations here. So these are 6 locations that we were also visited last year on Cayuga Lake, kind of throughout the water body there. And then a number of 5 or 6 Seneca sites on private boat launches, docks owned by Seneca Lake Pure Waters Association members and constituents. Again, those will be 50-meter point intercept grids every month, June through October, that we will be visiting monthly. This will be a team of 2 people primarily doing this work this year, so it's a little bit smaller of a team, and 1 fieldwork coordinator. That fieldwork coordinator is a new position for us. It will be generally split between the work that we're doing in the field with our early detection rapid response work, the point intercept surveys, and split between that and also helping with the watercraft steward program. So just an additional role, additional staff member to help kind of support those pretty time intensive and admin heavy programs, and as well as helping a lot in the field. So 3 of these Cayuga sites, of course, will be monitoring current hydrilla management areas. So we'll be working with the Cayuga Lake Hydrilla Task Force and New York State DEC. Emily Timke is here. We'll be working with them to make sure that we, are supplementing each other's work and complementing each other's work and reducing kind of duplication of efforts, of course. So just kind of continuing to work on this. A couple of the other modifications we made last year that we're going to be carrying over to this year is specifically with what data we're actually collecting. So typically the I guess effective data points that we collect for each rake toss is generally amounts to presence absence of a specific invasive species, where it is, and when it is. Essentially, we do collect somewhat subjective metric looking at how much plants are on the actual rake toss and kind of putting that into a category. We are still doing that, but what we're doing to add and kind of enhance our ability to quantify actually what we're finding on rake tosses, we're looking at species volume. So we're actually using, I don't have photos of it, I guess, but we're going to we use beakers to actually estimate the volume for each break toss, essentially, to have an actual continuous variable that we can use to learn more about what we're finding. And also we're looking at the percent, basically percent plant matter of each individual species, so we can get a closer idea of abundance of different invasives and natives in our different areas. So what this is going to allow us to do is, in addition to, there's all kinds of things that we can learn from this, but it's just going to help us learn more about how plants and plant communities interact and change over time, year to year, and also within the year, and also how these communities are different throughout the water body here. So obviously, Cayuga Lake is pretty big. It's got many different substrates, and kind of its different parts of the lake are subjected to different environmental impacts, including tributaries, sedimentation, different lake bed characteristics, and so on. So that's generally kind of what we're going for with our EDRR work here. So we're also going to be continuing our old, I guess, tradition of working on water chestnut. Of course, it still remains a priority for the Finger Lakes, but really for the central New York and kind of the region broadly. Part of the work that we're going to be doing this year with our field team will be leading water chestnut pulls, which will be focused on our small tech team, but also we're hoping to incorporate with volunteer groups and staff and members at the Finger Lakes National Forest. So this is part of a U.S. Forest Service, a GLRI project that's focused on inventorying invasive species and native species, and also collecting water quality data and terrestrial data on all the 83 ponds on Finger Lakes National Forest. So Josh is going to give a full in-depth report on that a little bit later, but just to give you an idea of like how we're following up with our first 2 years of work there, we've identified 12 ponds that have water chestnut. That's an increase of over 100% from when they last did a survey there. So I think when we got this data set, there were about 5 ponds that had water chestnut. So now we're up to significantly more. So our approach here is to just go and pull as much as we can and remove as much water chestnut from these ponds as possible. Of course, education outreach always being a priority for us. We're trying to engage, again, the public. So when it comes time, we'll be reaching out to our partner lists to kind of get people interested and come help out. It's a great way to just spend the outdoors, spend some time in the outdoors. Finger Lakes National Forest is beautiful. And you might even get to see a cow or two up close and personal, which is always pretty exciting. Okay, so our Watercraft Steward program, this is really one of our flagship AIS programs. Many folks in this room are well acquainted with it at this point. But what we are, this is, in many cases, this is business as usual for the Steward program. We're going to have at least 20 stewards throughout the region. This is a list of our sites here. I know it's kind of small, but hopefully big enough. So we have a pretty good distribution throughout the Finger Lakes, on most of the proper Finger Lakes, and also up towards other parts in Monroe County, and even outside of the watershed in places like Dorchester Park, which is down near Binghamton, like Whitney Point area, and then Juanita-Lamoka Lake, which is barely outside of the watershed, but is out. But we are going to be continuing work. We have ongoing agreements with Monroe County. So you notice some Erie Canal launches that kind of look like they stick out a little bit in terms of, why are we there ultimately? And they are valuable launches. I think one of the big things we're trying to approach with these, which is we're going to be there kind of on the guidance of the Monroe County Soil and Water Conservation District, who annually contributes a funding to provide additional steward resources in Monroe County. So they've identified a couple of launches as ones they want to learn more about. So we're gonna reach out to those locations and try to see what we can find to see if those are valuable to add to our kind of bigger list of launches that we cover in the region. We also have an ongoing agreement, or an annual agreement, with Canandaigua Lake Watershed Council to provide additional stewards on Canandaigua Lake at the north end, at the south end, and on Honeoye Lake. So we will be continuing that project. Part of that project also includes the operation of a decontamination unit at Canandaigua Lake. So anybody who's been there in the past couple of years has noticed the new big turnaround with a little hut and maybe a steward using what looks like a great big power washer. That is our staff, of course. They will be continuing that project this year. That is a collaboration between the PRISM, the City of Canandaigua, New York State Parks, and New York State DEC. So everybody's getting a piece of that agreement there, and we're doing a lot of good work. There's a lot of learning right now. The site was not designed to have a decon station there, of course, nor were many in our region, but we still want to try to incorporate that tool to continue to address invasive species in the region to a greater degree. So we are going to be continuing that. Okay. We're also going to be continuing some, in my opinion, exciting might be a stretch. It's exciting for me. Last year, we started a project with our watercraft stewards focused on skimming floating macrophytes on boat ramps. So this came from some research that we did, that Bill Brown did, primarily looking at where and how invasive species actually become hitchhikers. So is it on the boat, is it on a prop, or is it elsewhere? There was an old article from a research project from Canada in, I think, the early 80s or late 70s that basically found that was actually looking at hitchhikers in Canada that long ago. And what they found was essentially that it's really the trailers that are collecting the plants and not as much the boats themselves. So what we wanted to do is, we put our heads down and thought, how can we actually test this? So last year, what we did, and how we can address that problem. So how, we're not going to get rid of the boats necessarily, but how can we reduce the number of detections that happen on the lake? How can we reduce hitchhikers, the level of hitchhikers coming out of a water body through a boat ramp? So what we decided on doing was basically equipping some of our stewards with these basically just pool skimmer rakes or pool skimmers. They would do a set amount of effort for skimming the floating macrophytes within a ramp daily. And then after the fact, we were able to look back at the steward data to look at differences between detection rates on days where people skimmed and days where people did not skim. So the data is definitely early. We had some snafus with actually the rollout of this project, but the very preliminary data is promising that reducing the amount of floating plants inside of a boat ramp is actually going to increase or decrease the level of detections, thus, just making the steward's job a little bit easier, and even kind of possibly making it so the steward's job is not quite as necessary, even. Not that we're ever going to get rid of them, but it just helps stewards ultimately be more effective by actually actively removing these floating plants. So we're going to be continuing this year. So if you're out with a steward and they're look like they're cleaning the pool and the boat ramp, that is exactly what they're doing, pretty much. Okay, moving on to some terrestrial programming updates. Again, I'm sort of bearing the lead, but basically we're still working on transitioning from our previous terrestrial program management and even kind of the sort of coordinator level staffing for that, and still working to identify. So basically that has been a gap that we have had in the planning phases of this field season, but we have done our best and are still working on trying to come up with projects for our seasonal staff that we will ultimately hire to actually work on this year. So these are a couple of examples. We have additional kind of irons in the fire, but these are kind of the tangible projects that we plan on starting with this year and continuing to develop as we have time. So Keuka Outlet Trail, this is building on a project that Bill Brown did a number of years ago. It was actually, oddly, before he worked here, he received a PRISM small grant to actually do this work on his own through Keuka College and some students there. But what we're going to do, what we want to do is essentially test different management methods for wild parsnip. So in addition to the actual management and removal of it, we want to test what is the most effective way to essentially reseed and restore an area after wild parsnip treatment has completed. So this will ultimately involve setting up a number of test plots and doing different types of actual treatment and treating it using different methodology and using different plants and strategies to actually restore the area that's been where the wild parsnip has been pulled. Unfortunately, this year we ran into, so I'm sure many people in this room know, the updates to the New York State DEC's wetland permitting process, and this has unfortunately impacted this. We were just a little bit late in getting this rolled out. Like I said, staffing transition, blah, blah.
Sam, I have just a comment from somebody online who can't hear the audio very well. If the microphone is on, or if we could.
Test.
Sorry. That's
should be really bad, but it won't let me.
You can unlike that automatically adjust microphone. I just typed it in.
Does that work better? I changed my mic. Does that work better?
It says OMG, yes.
Okay, great. All right. Well, I'm sure you might be able to, I don't know why the recording would be any different, but maybe the recording's different when we're done with this. All right. Thank you for the heads up. If anybody wants any clarification on anything that I missed, I'm happy to go back. No problem. Okay, like I said, we are working with Friends of Cute Lake Outlet Trail on this project, and we'll continue to do so in the future. Spotted lanternfly, I was on the cusp of adding a couple of things to this slide because it's hard to remember exactly when the last time we met and things that happen in between. Spotted lanternfly, we actually were, I don't know if lucky is the right word, but we were actually the first people to identify, we as in PRISM staff, meaning Bill Brown and Ben Kelly, were the first people to identify spotted lanternfly here in Ontario County. It was right on campus, actually. So they were literally on a walk down to the lake, down to the boathouse right off of campus. And of course, they walked by a big pile of Atlantis, Tree of Heaven, very regularly. It's right along a Finger Lakes rail there, and they found a pretty significant population of spotted lanternfly. So with that, with the newer populations in, I think, Romulus or Ovid 2 summers ago, we are trying to match that effort and response and kind of what's happening here and anticipate new populations of spotted lanternfly in the region. So in addition to that, New York State Department of Ag and Markets, who are typically the lead guidance for spotted lanternfly activities generally have reduced their funding for spotted lanternfly, specifically for addressing spotted lanternfly. So we're, again, trying to fill this gap here in the region as a really high priority. So what we're going to be doing is identifying, basically using GIS existing data to sort of identify areas that should be monitored early. So we're going to be using public, so things like IMAP invasives and iNaturalist to be looking for tree of heaven locations regionally. We're going to be looking at things like rest stops, truck stops, rail yards, if they allow us to actually survey, which is unlikely, but we'll do our best. And other major transportation corridors and hubs to sort of triangulate where we want to actually monitor and set up traps for spotted lanternfly. We're also hoping to engage local vineyards in actively doing perimeter surveys, identifying trap trees as well on their property so that we can actively set up a really good, effective monitoring program so that we can detect as early as possible spotted lanternfly when it ultimately does start to spread to some of these other wineries. It was only in one winery at first, and now it's just on the railway. So there's no wine associated with this, but there's wineries literally all around us. So only it's bound to happen eventually. So that will be our spotted lanternfly work. That'll actually take up quite a bit of our time this year. We're really trying to focus on that as a major priority. The Hypena opulenta biocontrol project that we did last year to these are still in experimental stages, or not experimental, but they're early establishment stages, I guess I would describe them. So there's very few. So this is a biocontrol moth, and the caterpillar eats pale swallowwort. There have been very few successful applications of this where a consistent population was actually established through one of these tests. But we did one last year. We did have some success in that all of our moths lived and they ate a lot of pale swallowwort. This entire 10 by 10 grid here was totally full of swallowwort, and every single plant in there was skeletonized to a point where we actually opened up the window so that they wouldn't run out of food. And we're actually eating all of the swallowwort around it as well. So what we're waiting on here is to go back to see if any of our insects overwintered successfully. That would be a great step at establishing. This is in Mendon Ponds Park. We're hoping to reestablish this and relaunch this again this year in a similar or new location in the same area. With additional moths. We're also hoping to expand this program to buy another fence or another cube so that we can set up another population and possibly put it in a new area as well. So like I said, other projects TBD. We're going to be bringing somebody on here in the next couple of weeks and hope that we can pretty swiftly move into developing new programs. Could somebody let Tom in? Hey, Amy. Oh, he's in. Never mind. Sorry. Yeah, Tom, you're good. All right. Moving on. Good Lord. Okay, volunteer programs. We just wrapped up, or, again, we're I'm going to sound like a broken record. We've been short-staffed for a while. Our Hemlock Survey program, which usually is a very interactive program where we're actively corresponding with volunteers. We just haven't had somebody to be that role. So really our Hemlock volunteers this year were all stars. We basically were able to set it and forget it and still collect some decent data here. We pretty much did an initial training and said, we'll talk to you guys in a couple months, and still haven't talked to them again, but are hoping to soon. But they were still able to get out there and collect some good data. So found we had 23 volunteers active, 23 positive detections, and 63 non-detects for Hemlock woolly adelgid. Trail survey program, the timeline will be shifted slightly. We're hoping that that can be launched as soon as our terrestrial program manager comes online. So I'm hoping sometime in June that can be addressed. We will have registration and a registration survey, recruitment survey that's developed by RIT students to sort of gauge interests and look at preferences and other reasons for integrating and working with this program. So keep an eye out for that. Macrophyte Survey Program, we're essentially going to be launching it today. We're going to be taking, unfortunately, a similar approach to how we did our hemlock woolly adelgid program. We are going to essentially set it and forget it until we have somebody on staff that can actively monitor and interact with our volunteers. So we'll be training today. We'll be recording the training and posting it to the registration website where people are actually going to register and sign up for this. So we hope that people still remain active and can still do this without our input. And we're hoping to get back to everybody's inquiries that happen through the app later in the summer. A specific program update for hemlock woolly adelgid. This is another GLRI program partnering with New York State Parks. We just a general overview. We treated a couple thousand trees on 4 state parks in high priority riparian areas, steep slope areas in parks, including Watkins Glen, Buttermilk, Fillmore Glen, and one more that is escaping me. But we were able to leverage some PRISM dollars last year to actually pay for this. And it resulted in us being able to expand the program to actually expand treatment again this year. So we're working closely with parks to identify new location. The target area that we're looking at right now is actually hemlock trees in the main gorge. You can't see any hemlock here, but this is like the main gorge at Watkins Glen. This is a really high priority area for hemlock conservation in the state that we're excited to work with them on. An RFP for that work should be released later in the summer. Our education outreach has been chugging along. This year, we presented several posters and a presentation at the Northeast Aquatic Plant Management Society meeting in Saratoga in January or February. I can't remember. We also had our Finger Lakes Research Conference. We had over 150 people visiting here on campus over at the Scanling Center. We had tons of great presentations on everything from contaminants to nutrients and HABs, watershed management, and even invasive species. A bunch of our staff did some great posters that we had out as well, focused on invasive species. Here are a couple of them. I was going to go into detail, but I think I'm running out of time. We are excited to announce we have a manuscript in revision that's focused on watercraft steward data. This is the poster version, but basically we took watercraft steward data from 4 years and we're able to learn a hell of a lot from that data based on what we've been collecting with stewards. So it's a great step in the right direction for learning what programs can actually do with a strong data set like the steward program. More education outreach. We hosted a bunch of workshops, or 2 workshops, did a bunch of events. We're constantly involved in partner programs statewide and regionally. We did also make some pretty significant updates to our field guide and management guide. Updated versions are down here. We added some new species. We updated the appendices and the glossary, and also updated the distribution maps at the bottom of each location. So feel free to take as many of these as you want. If you have people that you want to give these to, we're trying to get rid of them. So please take a handful if you are so inclined. Looking ahead, we have a new logo. We are proud new owners of this logo here. This was done in accordance with New York State DEC. So this was developed right alongside by the same designers that updated all the other PRISM logos. I should have actually put those here. So this is basically part of a big package that is run by or kind of guided by the DEC. So all other PRISM logos look similar to this, as does the big, like the New York State invasive species kind of general branding. This fits directly in that branding program. So included in this logo, for those logo buffs out there, we do have, this is not just all random, of course. The new color, we have the strong winery and grape industry represented in the color here in this deep red. We, of course, have our forest lands here. our agriculture, and of course the lake prominent in the middle as all different specific parts of this logo. So that's that pretty much. We are in the final stages of contract execution to host the next Finger Lakes PRISM contract. So that will be between 25 and 28. It's a 4-year contract. And a lot will essentially come with that. Oops, a couple people waiting in the waiting room. Sorry. Oops. Okay. So with a new contract, this brings a lot of exciting opportunities for us to shift things. And of course, a big change this year for this contract will be kind of how we're operating our staffing. So with we have new staff positions that we're going to be able to fund through this new contract, and are excited about that. And of course, not what we're excited for. We're not excited about this, but we also do have people on our staff that are currently taking on some of these positions that are moving on. So I'll get to it in a second, but Amy is moving on, as is Bill Brown. So we are going to be hiring 3 new program managers. That's the aquatic manager, the terrestrial manager, and an education and outreach manager. One small tweak to how we're generally organizing this work is typically the AIS program manager is the biggest change, I will say, is the AIS program manager typically runs the Watercraft Steward program. We are now housing that program under the education and outreach program. So a little bit of a shift in kind of who's responsible for what. Ultimately, I think it'll go a long way in kind of creating more space for us to actually kind of do a lot of great work, kind of looking at aquatic ecosystems regionally. Also included in this will be strategic planning and website updates that we're hoping to engage our partners in, of course, within the first year here. So more details to come on that. Okay, finally, huge thanks to our partners and volunteers. We have a number of different funders, of course. And also just want to send a farewell to Amy. Amy's last day is next week. Amy was our AIS program manager for 2.5 years. So Amy is just leaving right before the beginning of the Steward program, but she's been invaluable in helping us prepare for it and just continuing this program for the past 2 years. So thank you, Amy, for all the work that you've done with us. I'm sure you'll be very pleased with your new work, and she's going to be can I say, you're going to be a freelance environmental illustrator. Thank you. We're very excited to watch you do that. So congratulations, Amy, and thanks for all the work. We're also saying farewell to Bill Brown. Bill is moving on to work down at Cornell at the end of this week. Bill has been a little bit less of a visual and kind of visible part of our work over the past 2 years that he's been here, but Bill has done a ton of work on the evaluation side. He's been the program analyst for us for 2 years and really has made some pretty indelible marks on the way that we do our work here, on everything from the way we're putting together reports to the way we're collecting data and how we're designing these programs. So Bill will be missed as much as Amy will be, of course, and he will be moving down to work in Baron Belassi's lab at Cornell. So that's what I've got. I don't know that I might have gone over time. Any questions about any of this stuff before we move on to our next content here? Maybe not. Oh, of course. New York State Invasive Species Expo is, what's the date? September 14th, 2025, Saratoga. This is actually a great event. They had it 2 years ago for the first time at the Saratoga Spa State Park. Obviously a great venue, but it's a great way to, it is free. So anybody can go to this. All you have to do is get yourself there and get a hotel. But it's a great interactive expo conference experience, I will call it, that focuses on the public development side, but also has very specific professional development opportunities and focuses for invasive species managers, land managers. Really, the application of this to professionals in conservation or environmental science are huge, and anybody in those fields really should consider going to this event. I'll be hoping to give at least one presentation. There's workshops, there's demos, activities. There's more classical, more conference talks, things like that. So really great event, very well done. I'm sure Tom can answer any detailed questions. Yeah, Tom.
So just wanted to add, there's also student posters.
Oh yeah.
We mentioned student posters earlier and
Yep.
and then the deadline for submissions has been extended.
Yes.
To May 16th.
Great. Yes. So for anybody that didn't hear, that's a great point. They're offering funding for student posters, correct?
There is some funding for student attendance, travel and hotel.
Great. Yes. So just Google New York State Invasive Species Expo. It's on NIAS.info. So check that out for more details. It just extended the portal for abstracts as well. So and that was my actual last slide. So all right, thanks everybody. I'm going to welcome Josh Neff now to come talk about our project on the Finger Lakes National Forest. All right.
There is a sign up sheet going around, if you haven't,
It didn't make its way all the way around.
All right. Thanks, Josh.
Yep. Thank you. So my name is Josh Neff. I'm currently the AIS Fieldwork Coordinator. Like Sam had mentioned, that is one of the positions we are currently hiring for. So I'm kind of shifting my role a little bit here soon. We'll still be involved with our aquatic programming, but we'll be kind of integrating some of our aquatic programming with some of the capabilities of our lab at the Finger Lakes Institute. So that's kind of where the shift of my position is going. But I'm still currently the field coordinator, and again, we'll be involved with a lot of our aquatic programming in the future. I think by this time, a lot of people in the room know who I am. I've been around the PRISM for 6 years now, which feels like a long time, but a short time at the same time. So this presentation here is our work in the Finger Lakes National Forest. So we received a grant from the U.S. Forest Service, essentially to survey all of the ponds in the Finger Lakes National Forest. As Sam had mentioned, there's 83 of them. Looking for or documenting aquatic invasive species. Kind of water chestnut is kind of the focus or the forefront of their mind, but we're also collecting other, native plant data, other invasive species data, terrestrial data, as Sam had mentioned as well, and then also some water quality data. So the Finger Lakes National Forest, it's located to the southeast of us here in between Cayuga and Seneca Lakes. It's a multi-use forest with ecological and recreational value to the Finger Lakes region. So, a lot of recreational value. There's a lot of hiking trails, ponds for fishing or stocked ponds for fishing. People will hunt in the Finger Lakes National Forest, but there's also a lot of ecological value, sediment retention, erosion control, things like that, specifically what these ponds are for. And again, there's 83 ponds in the Finger Lakes National Forest, pretty varying in size. The largest ponds, actually I can point. I'm not sure how to do that, but anyways, the largest pond up here is Teeter Pond. It's about 14 acres. And then many of the ponds are actually less than like half an acre. There's even ponds that are like less than 0.02 acres. So very small ponds, generally in the Finger Lakes National Forest. I'll get into it a little bit more, but we also did some kind of classification of these ponds. So some of the ponds are located in the forest. Some of the ponds are located in pastures within the Finger Lakes National Forest, so on and so forth. So there was kind of 4 major goals of this Finger Lakes National Forest project. The first of which, surveying the 83 ponds for the presence of aquatic invasive species, controlling water chestnut populations within those Finger Lakes National Forest ponds, and then also attending and presenting at outreach events, engaging youth about aquatic invasive species, but then also coordinating community science AIS monitoring program in the Finger Lakes National Forest region. So similar to, or exactly the same as, we're leveraging the MACFI survey program essentially for that 4th bullet point. So we kind of created these regimes of or sampling regimes within the Finger Lakes National Forest. So each pond was kind of assigned one of these 4 groups. And these 4 groups are kind of cascading in nature. So the first group here is just going and determining whether or not water chestnut is present in the pond. The kind of second group of sampling regime is AIS survey. So that includes doing rake tosses at each of these ponds that are within that group, but also documenting presence of water chestnut. Then the third group here is it kind of adds water quality into the fold there. So those ponds get water quality or water collected for analysis while also doing the AIS survey, so rake tosses and water chestnut presence absence. And then the fourth group there is folding in terrestrial plant data. Collecting plant data or terrestrial plant data around the perimeter of the ponds. And I will just kind of mention here, pretty early on in our sampling in the Finger Lakes National Forest, we started to kind of shift away from that first group there of water chestnut presence absence. We originally had designed these, again, in the interest of time. So we just didn't think we would have time to do like a full survey at each of the 83 ponds. We quickly realized that we would have time to do that, but we also just kind of realized that like some of these ponds are a little bit difficult to get to. You know, you have to hike half a mile through multiflora rows to get to them. So we just kind of assumed like we're there, we might as well do rake tosses while we're here. It's kind of a waste of time to like you get to the pond, look at it, no water chestnut, turn around and walk away. So we started to just add in the AIS survey essentially to all of the ponds that we visited. So the survey methods for doing the rake tosses at the pond. So we aim to do 8 rake tosses at each of these ponds. Ideally, we were doing them at each of the cardinal and ordinal directions. So north, northeast, east, southeast, etc., on and on. Some of the ponds were not accessible. We weren't able to walk fully around the perimeter for multiple reasons. Some of them were just, again, like multiflora rows or really thick vegetation that was difficult to get through. Some of them were just very like mucky. You couldn't really walk around the sides without sinking into your waist. So at those ponds, we just tried to do 8 rake tosses evenly spaced around the pond. So with each rake toss, the data that we were collecting were both the pond name and ID number. We kind of labeled the ponds with their own unique ID numbers just because some of the pond names are a little bit like, I don't know, kind of not all over the place, but like some of the ponds were named like C45-302, and then the next pond would be like C46-305. So just to kind of avoid confusion, we just made basically a unique ID number of each pond. And that's how we also went about like analyzing the data and just kind of to keep track of which pond was which. We also collected water depth where we were doing the rake toss. Again, small ponds, most of them were pretty shallow, less than like a couple of meters. We recorded rake toss density, so that kind of subjective density class of 0, trace, sparse, medium, dense. But then to add on to that, and again, like Sam had mentioned, and this was also a change we did with our point intercept surveys, was starting to collect macrophyte volume. So you'll see the 2 beakers here. This is a 500 milliliter one, and the larger one here is 2000 milliliters. So just to kind of help us quantify a continuous variable of how much plant material is coming up on the rake. And then with all the species that were identified on the rake, we would also record a percent abundance. So if we had, 2 species, was it a 50-50 split, a 75-25 split, on and on for the number of species that we observed. So then we also were pulling water chestnut from these ponds. So essentially we generated a list of ponds with water chestnut in them based on our initial survey of these ponds. So after our initial survey of 83 ponds, we basically went back, we realized that there was 12 ponds that had water chestnut present in them. So we essentially made that list and then visited those ponds to pull water chestnut, or at least a subset of those ponds. Again, we coordinated with volunteers to select water chestnut pull locations. We had 1 volunteer day essentially in the Finger Lakes National Forest to help with water chestnut pulls. And for that day, we selected Teeter Pond, which I alluded to previously is the largest pond there. But there's also a couple other reasons we chose Teeter Pond. One, it was just a little bit more of an accessible pond. There was a parking area with a trail leading back to the pond, which is beneficial for having volunteers there. So we carted our kayaks and canoes and the jet sleds down the trail to the edge of Teeter Pond and kind of let volunteers off. The water chestnut population there is pretty sparse. So it was a lot more of like a search and find type of pull where we're paddling around the perimeter. You find a couple of rosettes, you're pulling those, making your way along, searching along, mostly along the edge. There were some, rosettes in the middle of the pond, but generally around the perimeter. So again, a little bit more of like a search and find, a little bit more engaging than I know most of you are probably aware of, like sitting in a monoculture of water chestnut and just nonstop pulling up rosettes and filling up your kayak or garbage bag or jet sled in our case, one after another. So it's a little bit more engaging and a little bit more of a fun pull for volunteers. And once the water chestnut was removed from these ponds or from Teeter Pond, we basically made a big pile of it, used these bins to weigh how much water chestnut we had removed, and then to dispose of water chestnut right on site. So we had permission from the Finger Lakes National Forest that we could just make piles and dispose of, kind of back away from the pond. And then, do I have another slide? No. And so that was Teeter Pond. We did the volunteer pull at. And then we had basically 3 other ponds that we had time to visit this year to pull water chestnut from: Ballard, Bumpus, and Burdick Stock Pond. Those ponds were kind of just like fit in on extra time. So there was days where we finished the subset of ponds that we wanted to survey early. So we would just send the field team to those 3 ponds and they pulled the water chestnut that were in those ponds. Again, pretty similar to Teeter, where they were pretty sparsely populated. Those 3 ponds in particular really only had, we didn't actually weigh the water chestnut just because we didn't have the material with us to do it, but they were all like less than 10 pounds most likely. But we were able to like fully remove the water chestnut from those 3 ponds. And so again, that's basically, so 4 of the 12 ponds that had water chestnut, we were able to visit last year and remove the water chestnut. I will be talking about this a little bit later in another slide, but we will be revisiting some of those other ponds that we weren't able to visit last year to remove water chestnut. So our water quality survey, essentially we had kind of 3 different things that we were doing there. One was using a Manta probe to determine some kind of basic water quality parameters, so dissolved oxygen or DO saturation, specific conductance, pH, and temperature. And then we also collected water in bottles for analysis at the lab at the Finger Lakes Institute. So we did filtered water essentially for the top 4 bullet points there. So SRP, soluble reactive phosphorus, nitrous oxide, ammonia. And then we collected water in various size bottles like you see in the photo there for total nitrogen, total phosphorus, bromide, and chlorophyll. And then lastly, we also took a Secure reading to determine water clarity. And then the kind of last sampling regime that was included in the pond survey was terrestrial invasive species survey. So similar to the kind of rake tosses, we used the cardinal directions to determine where these quadrats were being put down. So there are 10-meter quadrats in the 4 cardinal directions surrounding the perimeter of the ponds that were prescribed for the sampling regime. With the terrestrial survey, our terrestrial invasive species tech team were the ones who carried out this survey, and they recorded presence absence of woody herbaceous and graminoid invasive species within that quadrat, while also assigning a % cover for each of the individual invasive species that they observed. And they also did record native species when identifiable. I know they ran into a lot of problems with like grasses essentially being unidentifiable, but we're still generally classified as like graminoid species or whatever class species they're in, but maybe not identified down to species. So some of the results from our survey in the ponds, we saw water chestnut in 12 of the 72 ponds sampled. So that 72 number is different than the 83 total ponds I've been kind of touting. The reason we surveyed 72 of the ponds was just some of the ponds were, again, not accessible. So either like having to go through really thick cattails or multiflora rose was a really popular one. Or there's also some ponds that were just not existent anymore. They had filled in essentially with sediment and were no longer a pond. So within those 72 ponds that we surveyed, we observed 18 aquatic species. I guess you probably can't see that table at all, but essentially the table was really just, I wanted to show the top row so you could see which species we're finding in the Finger Lakes National Forest. But I guess the two common ones, and probably not a surprise to many people in here, but the two common species that we were able to, or the two most common species that we were observing were Elodea and Eurasian water milfoil. And that was both by volume and detection. So essentially those were coming up most often on the rake, but also while they were coming up on the rake, we were getting the most plant volume of Elodea and Eurasian water milfoil. And then just kind of an interesting little tidbit, we happened to look at the ponds that had water chestnut within them and which species we were finding in those ponds. Same pattern held with Elodea and Eurasian water milfoil. Those were, again, the most abundant by volume and detection number. But we saw something kind of interesting with coontail. Coontail is, again, a common species and was one of the more common species we were finding in generally all of the Finger Lakes National Forest ponds. But we kind of saw a drop off in the proportion of coontail in ponds with water chestnut versus ponds that had no water chestnut. So the water chestnut removal results. So again, 4 water chestnut removal events last summer. The volunteer poll at Teeter Pond. I guess I kind of got ahead of myself a little bit on the method slide, but the volunteer poll at Teeter Pond, and then we also did our 3 AIS tech team led polls at Ballard, Bumpus, and Burdick Stock Pond. So Teeter Pond, we had 7 volunteers show up. Some of the volunteers were like Schuyler County Soil and Water Conservation District. We even had volunteers like within our own steward program that were there on their own time for the water chestnut poll. And in total, we were able to remove 89 pounds of water chestnut from Teeter Pond. And again, I'm repeating myself a little bit, but the Ballard, Bumpus, and Burdick Stock ponds, we didn't weigh them. We estimated less than 10 pounds removed per pond, but we were able to completely control the water chestnut populations that were in the pond last summer. So some of the water quality results, and I kind of want to preempt this slide by saying like a lot of these analyses are still in progress. We're still kind of working towards a final report, but these are some of the things that we're looking at when it comes to water quality. So first of all, we wanted to look to see if there are any water quality parameters that were predictive of aquatic invasive species being present in the ponds. And that initial analysis kind of came back and said, no, there's not really any statistically significant water quality parameters that are predictors of aquatic invasive species being present. Although you can, in this table here, you can kind of see TP and DO, which I have starred there, did exhibit, kind of some positive relationship with the presence of aquatic invasive species. And then we also, so that was, looking to see if there was any predictive predictive water quality parameters that will predict the presence of aquatic invasive species. The second graph here on the right is looking at if there's differences between ponds that are in forest land use or pasture land use. I'll get into that a little bit more in my next couple of slides, the pasture versus forest ponds. But that is some of the other things that we're kind of looking at with the water quality data. And it might be a little bit difficult to see, but this is for forest ponds and then this is for pasture ponds. So it kind of makes sense. Total nitrogen is a little bit higher in pasture ponds. The forest ponds surrounded by trees, surrounded by vegetation, they have a little bit more, nutrient control surrounding the ponds. And also the pasture ponds are mostly cow ponds. So you can imagine the nutrient input that the cows are also putting into the ponds. So terrestrial survey results, there's 10 ponds total surveyed for terrestrial invasive species. Within those 10 ponds, or I guess surrounding those 10 ponds, there were 14 terrestrial invasive species identified, and then 65 native species, which were mostly forb species. And I guess as I have mentioned previously, the multiflora rose was present at all 10 of the ponds surveyed. Again, was one of the very common, we were seeing that all over the place in the Finger Lakes National Forest. But bush honeysuckle and common buckthorn were 2 of the other prominent terrestrial invasive species surrounding these Finger Lakes National Forest ponds. So this is kind of where I get into the pasture versus forest stuff that I had previously mentioned. But so this is looking at essentially the characteristics of the Finger Lakes National Forest. Are any are any of those characteristics predictors of aquatic invasive species? So the two that we looked at in particular so far are distance to road and pasture versus forest land use. So the distance to nearest road, we essentially, that was after we had collected all of our data, we essentially used ArcGIS Pro to measure essentially a pond edge to the nearest road. And we did find that the likelihood of detecting Eurasian water milfoil was negatively related to the nearest road. So essentially, the further you got away from the road, the less likely you were to find Eurasian water milfoil in that pond. You can kind of see there's the cluster of ponds at the top there. Those are all those points essentially represent a pond that had Eurasian water milfoil present, and all of those ponds were less than 250 meters from the nearest road. So, a possible explanation of that is that those ponds are near to edge habitat, which is can be a facilitator for invasive species presence, but also just the kind of accessibility aspect of that there's people have a little bit more accessibility going in between there, but also animals as well. And then the pasture versus forest pond. So looking at whether one or the other was more susceptible to having aquatic invasive species. And again, we kind of quantified this mostly using ArcGIS Pro, but there was also, some kind of field observation as well, whether you're standing in the middle of the forest or in the middle of a pasture. So we found that pasture ponds were more likely to contain Eurasian water milfoil. That is the figure on the top of the page. And similarly with curly-leaf pondweed, that ponds that were within pastures were more likely to have curly-leaf pondweed present in them. And similarly to kind of my last point, that likely due to being more accessible within the pastures and also other animals, but also the pasture ponds. So the Finger Lake Central Forest has the hectare grazing or an agreement with the Hectare Grazing Association. So cows are constantly rotated through pastures within the Finger Lake Central Forest. So you have cows moving in between ponds, which could potentially be a facilitator of invasive species spread, because we did observe multiple times. I mean, the cows would go right into the pond to drink. So there's some potential for spread with that. But again, other animals and also just the human pathway of movement. So the kind of last two goals of this project, community science and education and outreach, we essentially facilitated a community science program, again, with Amy's Microphy Survey program, where volunteers were collecting rake tosses we're collecting aquatic plant data using the rake toss method in the region. There was a total of 56 volunteers, 10 of which were within this region. So we use Seneca and Schuyler County, which are the surrounding counties of the Finger Lakes National Forest. And then for the education and outreach goal of this project, we attended multiple county conservation field days. I believe it was Cayuga and Ontario County. But within those county conservation field days, we were able to engage with over 1,300 youth of the Finger Lakes region. So our plans for 2025. So again, we will revisit the 12 ponds that we determined to have water chestnut populations through our surveys in 2024. So there will be removal events at 8 ponds essentially the 8 ponds that we didn't visit last year. We'll be revisiting again to remove the water chestnut. And then of the 4 ponds that we did visit last year, we're essentially going to do a monitoring event where we go back to that pond and see if the water chestnut has kind of popped back up. More than likely, I'm sure there will be some water chestnut or possibly rosettes that we missed it from the previous year. So we'll just be kind of monitoring that. And then if we do find water chestnut at those 4 ponds, we will be removing it. And then the kind of last thing, once we do finish up, that'll essentially be probably like 1 week of work visiting those 12 ponds. And then once we are done with that, we'll be kind of generating our final report for the Finger Lakes National Forest Ponds project. And I believe that is my last slide. So if anybody has any questions, but I just want to thank the U.S. Forest Service for giving us this grant. I guess I maybe should have mentioned this off the top, but a little bit of background on that. So this had been something that was going on for quite a while, although the last time that all of the ponds in the Finger Lakes National Forest had been surveyed was 2015. So you're looking at 9 years later, essentially, or a 9-year gap between surveys of the Finger Lakes National Forest ponds. So, so we kind of stepped in or received this grant and surveyed all of the ponds. And, really the main point of this grant is so that we can inform the Finger Lakes or the U.S. Forest Service. There's a fly flying around here. Anyways, inform the U.S. Forest Service on some of their management practices in the future. So not only regarding invasive species, but things like erosion control and nutrient control as well. So yeah, I will take any questions if anybody has any, and thank you for your time. Yep.
Can you describe in more detail how water chestnut is spread?
Yep. So they have the nutlets essentially. Oh.
Can you just repeat the question?
Oh yeah. Yeah. So the question was essentially just talking a little bit more about how water chestnut spreads. So water chestnut forms, it's a floating macrophyte. So it forms a rosette, a floating rosette that is on the top of the water surface. And underneath those rosettes, they have nutlets, they're called. So I'm sure people are somewhat familiar with those, but they have sharp spines on them. And so they hurt if you step on them. They're a little bit, they're sharp. If you even just grab one the wrong way, it'll kind of poke you. But anyway, so those nutlets form underneath the rosette, and then they'll drop off usually early or mid-July, somewhere around there. And then those rosettes will those nutlets will form roots, and then new rosettes are formed. So each rosette will have anywhere from 4 to 5 nutlets on it. And then each nutlet, when it's sprouting, can create anywhere from, 5 to 10 or so rosettes probably. So you can quickly, I always say this when we're out doing water chestnut pulls or with volunteers, you're pulling water chestnut and you kind of start to do the mental math of how easily it's spread just based on numbers of nutlets on each rosette, and then how many rosettes are formed from a nutlet. So, but yes, that is generally how they're spreading through their nutlets.
Those are attaching to people and livestock.
It is a possible vector of spread. The spines are barbed, so they'll stick right on waterfowl fur and things like that. But yeah, I guess human spread could also be one, but I think new populations, I think generally are probably attributed to waterfowl. But once it kind of takes control of one area, it pretty rapidly can spread and form a monoculture.
Yeah, also things as it's floating, things like fun can actually push it and spin carrots as well. So it's the nature of the invasive species is that it's spread often.
So do you take the precautions when you're surveying different ponds that you aren't spreading it?
Yep. Yeah, that's definitely kind of one of the things that's built into the routine of our field teams is, and similarly to the steward program, after we're going into a water body, or I guess before we're even entering the water body, we're usually kind of checking over all of our equipment, all of our kayaks, paddle, any of the equipment that we have. We're kind of doing a visual inspection, making sure that we're not carrying any invasive species with us, because we are pretty often moving between water bodies. And then once we're done for the day, it's kind of just built into our routine that we're wiping down and visually inspecting all of our equipment to make sure there are no invasive species fragments on them. Yep. All right.
Questions from the chat?
All right. Well, thank you. Yep.
All right. So as with any good PRISM meeting, partner meeting, we like to hear from our partners, of course. If it's okay, I'm just going to continue to record this so I don't have to actually write down everybody's updates. If anybody has any objections, let me know, but I'm going to do it unless I hear an objection. So I think now is a good time, like I said, to just I know some people signed up to give updates. It's a general, open call. You don't have to give updates. You can also just say hey and introduce yourself if you're new to the group as well. So I'm just going to go. Any calls for opener? Anybody want to start? What about you, Tom? I know it's your job to be here, so you have to give updates, right?
So I don't have any real great news.
That's okay.
Things going on at the federal government level. State budget, at least for many of our programs, is greatly reduced or reduced. Some budgets, like for Asian automobile, and we shape the next year and a half. While other things that are more impactful here, box pre-moth, spot lamp supply, there's minimal to no budget at all for those items, so we won't be able to support trapping as far as flying traps or any of that. However, if there are traps that are still usable and PRISM partners want to put those out, that would be fine. And also, if you can capture that data, we do have a new survey 123 data form for 2025. I can share that. I would just need whoever's entering that data to send me your ArcGIS account, and I will forward it on to Mike Formicelli, again, all the data stuff within that industry, and he will get you the updated form. Because along the same line, there's no outreach material available. I've had multiple requests recently, and we have nothing to give out. We have really very little to use in-house. And there's likely not to be any printed or produced soon. However, I do have PDFs, a lot of the printed material. I can show those. If you needed, 5 or 10 trifold, you take them down, you can pick those out. Certainly not cases and things. And the water bottles, tote bags, and all that are definitely no go.
Right.
I have a question about the materials like available online.
None of it is available.
Or no, the PDFs that you have,
Yeah.
I'm sorry, have any of those been updated with the new like spotted lanternfly distributions?
None of the spot electrified distributions were directly on those materials.
Okay,
It was links, and those links are updated frequently. That was by design, that way we were constantly throwing out outdated material. But the links are up to date. It's basically the Cornell integrated pest management site for spotted lanternfly. That's the best go-to. Our website gets updated with very vague kind of information. The more detailed stuff, especially what pesticides can be used for spotted lanternfly, that's all housed through Cornell IPM. And they have a specific site, page for spotted lanternfly, as does use the box pre-moth. So those resources are still available up to date, but the printed stuff. Yeah. A lot of it was buzzing around before.
it's
Migrated back here now. The only other thing would be, we do have some seasonal assistant horticulture positions available. So depending on the program, really, we do have some funding for some of that.
Okay.
But even a lot of that has been cut back. So if you do know anybody, especially anybody that's been impacted from the federal reductions, because USDA plant protection quarantine just for New York State has lost 22 people so far. That's statewide, program-wide. And none of it makes any sense. The SLF staff they had out here were let go. The SLF staff they have at JFK in New York City, they're still working. None of it makes any sense. I think the people making those decisions are not within New York, and they don't understand much of anything that goes on with PPQ, or us, or anything. That's my own personal opinion.
It unfortunately is being recorded, but
That's why I'm saying it's my personal opinion.
understood.
But but it's definitely having impacts industry-wide, statewide. So we are having some issues there.
Yeah, so just to retroactively introduce this Tom Algeier from New York State Department of Ag and Markets for anybody that didn't know. So
Sorry.
that's okay.
You know me, so sorry.
Yeah, of course. No, that's okay. Okay, I will also say on the topic of education outreach, and I'm wondering if Ag and Markets promotes, the DEC made a pretty solid one pager for spotted lanternfly. It doesn't have a distribution, but it actually is like up to date and in kind of its suggestions, identification, lifestyle or life cycle information. I don't know where you can find it. We can share it with you. We haven't printed, but also the DEC made it available somewhere. So just a general update that's available. Also, and not to put you on the spot, but just kind of, I mentioned earlier in my talk, we were talking about kind of us addressing kind of those cutbacks, and we're trying to expand. I reached out to you about, monitoring on rail stations. We'll see how that goes. Probably won't go.
That's a challenge.
But
for us.
yeah, but we'll try to just generally going to try to fill the gap as much as we can. And since we found it here on campus last year, we're really trying to make an effort to enhance that. So hopefully we can pick up, and we'll still plan on kind of going through the same, reporting processes with you guys. But just to sort of maybe jump topics. This is not just the Tom Algar under a microscope, but in terms of managing, and I think people just are interested in managing active populations of spotted lanternfly. From my understanding, the state isn't there's no resources to actually manage them. It's basically still monitor so that people can understand when it's coming to them and how to respond, and if they can anticipate things. Is that kind of generally still the company line for New York State generally?
We had some funding that was available for internal treatments.
Yeah.
We had pesticides that were available, golden pest spray oil.
Yep.
But that window of opportunity is come and gone. It's like late March, early April.
Gotcha.
It
For
targets
eggs.
big masses.
Yeah.
We did try doing some other treatments in previous years, but currently funding for the people that are doing those is not there. We geared up, we have our own in-house trained staff, do treatments, and there's no funding paid them to do them. It's pretty frustrating.
Right.
We are focusing on agricultural production areas. Obviously, the Finger Lakes, the west part of New York, and even Long Island on the east end, there's quite a bit of great production there. That sometimes gets forgotten about. So the best we can do at this point is we're giving the resources through Cornell IPM of, keeping up to date on what pesticides are effective, not just what pesticides, but also applications. Because spotted lanternfly is most active between 10 in the morning and 2 in the afternoon. So most pesticide applications in vineyards occur early morning and evening when the pollinator is not out. So that complicates things, and it also greatly increases labor and fuel costs for treatments. So all those things are kind of being, put into play.
Sure.
So they are looking for other pesticide options that can be used at different times of the day, that kind of thing. But again, the Cornell IPM list is the go-to for those. We're not abandoning SLF,
Of course.
but we're just doing what we can with what we have, and we have less than.
Yeah, it's obviously also extremely costly to manage these major populations. So anyways, thank you, Tom, for your insights.
Spread at this point too.
Yeah.
So stopping it from spreading 5 counties versus within a county, there's
Sure.
one more effective.
Question?
Kind of changing gears.
Yeah, of course.
So I'm Maura from Genesee Valley Conservancy out of Geneseo, new to the Conservancy and Stewardship Director role, but I have a question about a program we're launching this summer.
Sure.
Which is, an invasive species strike team, specifically on invasive species. We're hiring about 6 people, and I'll do a shout out to Kevin's coworker in the back. He's been at the Conservancy about 3 years and went from creating heat maps of terrestrial invasive species on all 5 of our preserves, so did the quadrants and all that, and now we're deploying 6 seasonal staff. But one residual question we have from the work planning is what to do with the biomass. So since it's the inaugural year and a lot of the staff will be folks that are currently in a bachelor's program or recently graduated, we're doing mechanical removal this summer. And a lot of the heavy hitters, buckthorn, multi-floor rose, honeysuckle, some periwinkle and such. But what Kevin did a lot last year with weekly volunteer groups was removing a lot of the buckthorn in trash bags. And taking to landfill, some piling and trying to get the roots right off the ground. But we're still kind of stuck on this increase of, we're gonna be increasing the biomass that we're removing and obviously don't love increasing the mass at landfills. So looking for any tips, advice on kind of getting around that. We've obviously considered some burning, but that's not a skill we have in the house. So here to take any advice.
And I will say that I would have to do some research to give like a better informed, I don't want to give bad info off the bat, but I'm glad you're considering it. And there are definitely options. And we just put together a pretty detailed management plan for buckthorn for one of our contracts for New York State Parks. And I know that there is a section in there about just disposal, but I don't have it off the top of my head, but we can connect afterwards unless anybody in the room has firsthand experience. Great. But otherwise I'm happy to advise kind of, sometime this week or next week.
Awesome. Thank you.
I'll just say we used to, like the wood chipper.
Okay. Yeah.
And before it goes to seed, obviously,
Sure.
we don't wanna like chip the seeds and then, yeah.
So that's just like, would you rent one or you have?
Yeah, yeah, you can rent one, yeah. You do definitely though, you're having seasonal staff, like, chippers are dangerous.
Yes.
You may even wanna like, again, I don't know budget wise, like you can contract that part out just for a safety perspective, cause yeah, I had to do, it was like a 2-day training of like throwing things in a chipper. So.
Yeah.
No, that's great advice.
They're very dangerous, but yeah, good to get rid of large piles of stuff.
Yeah.
Great.
And then would
Thank you.
would you use those wood chips or would they take it off site?
You can, I mean, we, like in a project we did, it was corn, some honeysuckle, we chipped it. And then we just used it to like kind of remulch a trail that was like right there, so, yeah.
Thank you.
Yeah.
Okay, anybody else would like to give updates? I know we have some SLIPWA folks, we have some other Lake Association folks.
But I don't care about.
Please mute, please. Hi, Jennifer.
Brandon has a question. Is there going to be any?
Yes, agreed.
Thank you. Yeah, I can see this. Thanks, Nadia. Yes, sorry, I didn't mention this. We are going to be having our in-person watercraft steward training. We do that every year and open it to the public. We train our own stewards. We also give our training to our aquatic invasive species technicians as just a primer for our background, PRISMs, everything in between, leading to a more detailed plant identification and ecology training as well. That's going to be Monday, May 19th through the 21st. I think all of it is going to be in person. It's going to be in this room. I think Amy's hopefully about to send out an email about it, maybe like today or tomorrow or something like that. So more details to come. So we'll probably put that out to our general partners. We can also send it to the list of people that are the attendees today here as well. Thank you for your question, Brandon. Yeah, Mary, please go ahead.
Hi, I'm Mary. I'm from Washington Manager. I just wanted to mention to the group that we've got our annual report from our Canisius Lake Watershed Council coming out. It should be up on their website. We have a portion of that dedicated to invasive species. We're in the middle of an update to our Canisius Lake Watershed Management Plan. It's been a 2-year project. We're about calendar wise, about a year into it, it feels like maybe we're a little bit farther ahead than CAP. We do have a portion of that also dedicated to invasive species. Luckily, we don't have some of the heavy hitters like hydrilla and water chestnut. But what I would ask in this group is if anybody sees something on the horizon, our plan goes for about 20 years. And so, for example, like it didn't mention apps 20 years ago, didn't know about it, wasn't in there. We're like, why didn't you mention it? So, well, we didn't know. So if you see something invasive species, species or a new emerging technology or anything in that category, you think, hey, maybe we should include it in our plan and address it now. I'd love to have that input. And a couple of the people in this group would be part of the reviewing body. Sam gets probably way too many emails from me, but we will have an opportunity for anybody who wants to here review our watershed management plan. Thank you.
Thank you, Mary. Anybody else would like to share, introduce themselves? Anybody online? I know we have some folks looking for, yeah, please.
Hi, I'm Stacey Bergel with
Oh, great. Stacy, thanks for coming.
Nice to see you. So I just wanted to share, I work in partnership with the DEC to manage the NIAS.info website, which, as I noted last meeting that I attended, has undergone a pretty dramatic update and facelift. It's got new accessibility functions. And the DEC and I are going to be releasing a joint press release for NISOS. So if you would like to have any events added to our calendar in advance of that press release, get those to your PRISM coordinators or reach out to me sooner rather than later, because that press release is probably going live next week. But even if you missed the press release and still want your events added to the new calendar, please get those up there.
Great. Yes. Nice. Thank you.
I'm still in denial that it's May already.
Yeah. No kidding. You're telling me. Okay. Anybody else? Yeah. Emily, please.
I can just do a quick update. So I'm Emily Benzinger. I am the Aquatic Invasive Species Coordinator for DEC Region 7. So kind of like the Eastern Finger Lakes, like legally to the east. So we will be working on some hydrilla management and monitoring on Cayuga Lake this year. So some of that monitoring, we're gonna again work with Finger Lakes PRISM. And like Sam mentioned, just make sure that we're not duplicating too many efforts, but we're still having pretty thorough coverage of the lake. And then we'll also be doing some management for hydrilla on Cayuga Lake. So that will include the South Aurora area, the Long Point, and then also south of Long Point State Park. So it's kind of on the eastern side of the lake. And then the Lansing, Ladoga Bay area, which is kind of southeast of the lake, and then the Sheldrake area, which is on the western shore, near like Ovid and Seneca County. We will also be doing some monitoring as well in the northern area, so Cayuga Lake State Park, the Island area. And then we're planning to work with Army Corps to do some joint survey efforts this summer and fall, and Finger Lakes PRISM also typically helps out with those. Some other hydrilla stuff that we're doing. So we're going to do some eDNA work down in the Susquehanna River. So we do have a really teeny tiny patch of hydrilla down at the Owego Creek and Susquehanna River where they kind of meet. So we're going to try and do some eDNA work to hopefully find out that it's just in that area. So we'll be doing that this fall. And then for water chestnut work, we'll be doing some removals with the Region 8 wildlife staff at the Montezuma Wildlife Management Area, so kind of on the Seneca River. And then also delineating some water chestnut in the wildlife management area. So it's kind of like there's like an island in the middle of the river, and then within that island there's some smaller ponds, and so we're going to do some delineation there. As well as hopefully work with our environmental conservation officers in the region to do some drone work and survey the water chestnut using that, because some parts that wildlife management area are pretty accessible by boat or by foot. And then we're also planning to do some macroinvertebrate monitoring with the New York State Water Resources Institute with Cornell. On the Seneca River and Cross Lake area. So we're gonna be monitoring inside and outside of water chestnut beds. And then also planning to do some submerged aquatic vegetation monitoring in some pre and post water chestnut management areas as well, kind of looking at some of the different management methods that are used. And that'll also be like in the Seneca River, Cross Lake area, and the Oswego River. And I think that is most of what I have. So,
Thank you. That sounds cool.
Yeah.
Excited to see that water chestnut management.
Yeah.
That sounds really cool. Great. Anybody online? I have Michelle Wunderlich, Emily Fell, anybody else that would like to give some quick updates? If not, we can move right along.
I don't have any updates, but I just wanted to introduce myself to everyone.
Sure.
My name's Gabriel Yerdon, and a couple of months ago, I joined the Finger Lakes Watershed Program with DC.
Oh, great.
And I replaced Amy Cameron as the environmental program specialist in our office. So just wanted to get my face and name out there, and if there's anybody that I can help or be involved in the program, let me know.
Great. Thanks for coming. Nice to meet you. Welcome to the Finger Lakes, I guess. Yeah.
Linda Sampson, Seven Lake Pier Waters Association. We have Hemlock Woolly Adelgid plan in place. We are going on private lands, perennial streams, assessing hemlock trees for the severity of the adelgid and to see if they could be treated. We have some funding in the works. And hopefully, if that comes through, we can start treating in fall. Right. Our water quality, water quality, Mark Petzold is going to sample more streams this year for flow. He is installing flow meters on several streams, and we're going to sample more streams for invertebrates.
It's great. Great to hear. Thank you. Anybody else? Last call. Sure.
Hi everyone, my name is Jayla White. I'm the Black Invasive Species Coordinator for Region 6, so up north. We'll just give a quick overview of what we're going to be doing this season. So I'm supposed to be running the CeeLo PRISM Woods program this year. That went out for bid, and unfortunately we didn't get any bids, so the program will not be happening this season. But we're hopeful for next year, and we're going to advertise it longer, and we'll see what happens. So this is a startup in our region, currently in the process of purchasing all of our field equipment and preparing for staff onboarding. Our first project, since we're up north, it takes a little longer for the plants to grow. We're going to be doing some mute swan management with egg oiling, marking up nests, and then we'll be working with the Region 6 wildlife staff for leaf removals.
Is this a PRISM meeting?
Yes.
It is, oh my God, never would have found it.
Oh, we need to sign it.
Thank you so much.
Sorry, please continue.
We are also doing a pre-dam level macrophyte survey at Utica Marsh Wildlife Management Area. There's a water control structure there that is damaged, and sorry, now the fly likes to go after. So we're gonna be doing a survey there before the dam is removed, and then we're gonna monitor it after to see what's gonna be growing in place of mostly the water chestnut that is totally infested the area, like this one. We are also going to be doing water chestnut removal and delineation of water chestnut infestations throughout Region 6 and some in Region 7. We're also going to be hosting our own water chestnut poll at Lakeview Wildlife Management Area on July 22nd with the rain date of July 24th. We will also be assisting Save the River and their water chestnut pool on Delton Bay and Sligo PRISM up at the Oswegatchie River. Then we are excited to start doing some research this year. We're going to be working with the DEC and the Water Resource Institute of Cornell to do some pilot waterfowl research on if waterfowl are zoochorinous vectors of aquatic basic plant dispersal throughout New York State. So this is going to be a pilot. It's brand new to us. Reading the protocol and doing this research. So none of that has actually happened yet, but we're hoping to collect any fragments or plant seeds, and we're going to identify those, and then we're going to see if they're still viable after passing through the gastrointestinal system. So very new, and have a poster about that and the expert. So we have some of that work actually done and built.
Neat. All right, thanks everybody for giving updates. I will be compiling some of these. Hopefully the recordings went through, and I'll be sharing and posting the minutes for this. So that concludes the main portion of our partner meeting. Thanks again, everybody, for coming. You don't have to leave, but I am going to be passing it over now to Amy Slentz to run our first and possibly only macrophyte survey program training of the year. I know we have some people online. Oh yeah, a bunch of people that have just been admitted. Great. So I'll just kind of address folks here once they get on, but thanks everybody for coming. And again, you're more than welcome to stay, but we're going to be a very specific citizen science macrophyte survey program reporting. So, all right, everybody online. Hi, thanks for coming. I'm just about to pass it over to Amy Slentz to run the macrophyte survey program training for the day. So thanks. Thank you, Amy. Let me open this up for you. Oh yeah, nice. There it is.
There it is. Okay, hi everyone. My name is Amy Slentz. Maybe I'll just give a slightly long hello to make sure the volume is good online. This one's
Yes,
great.
but I think if you want people to hear you so that we can do the recording.